Hi guys, welcome back to our channel and welcome back to season 2 of Active Concepts Masterclass Series. My name is Meg Mullins. I am a technical marketing professional here at AC. Last episode, we learned all about the dermal microbiome immunology assay with Morgan and Dan. But today, we are here with Grant Tyler, our R&D product development manager. Grant, it is great to have you here. Could you please, for the people, tell us about your background?
Yeah, hey Meg, thank you for having me. I'm excited to be here. So I graduated from the University of North Carolina at Chapel Hill with a degree in physics. So after graduation, I discovered an opportunity here at Active Concepts and discovered how cool cosmetic science is working in the R&D lab. So now, almost 3 years later, I'm still here as the product development manager, and I lead our team of R&D scientists to create exciting new active ingredients to be used in skincare and hair care formulas.
I love that for you, and I'm excited to learn about something new today. It's one of our studies. It's called the Intracellular Calcium Assay. So, Grant, why should we care about calcium?
Well, because our skin literally depends on it.
Wait, depends on it, really?
Yeah, our skin cells, or keratinocytes, make up our skin. These skin cells must do 3 things to protect our skin barrier's integrity. Would you know what they are?
I know what they are. I watched our season 1 episode all about skin biology, so I know that keratinocytes must proliferate, differentiate, and migrate.
Yeah, you got it. Did you know that those 3 things are dependent on intracellular calcium levels?
Now it makes sense. So intracellular calcium levels are really what's determining our skin barrier's integrity.
Absolutely. So intracellular calcium plays a key role in skin health and immune response because it can help reduce inflammation, which has a direct effect on tissue repair and keratinocyte function. Calcium also helps maintain skin moisture levels and promotes the production of antioxidants.
I always knew calcium was really good for our bones, like our parents tell us to drink milk, but I didn't know it was so important for our skin's health as well. So does that mean we all need to start putting milk on our skin and like taking milk baths like Cleopatra?
Not exactly. See, the way that our skin cells recognize calcium is not as easy as just applying a source of calcium externally. Our cells are dependent on ions like calcium.
Oh, well, I'm just personally glad I can keep my oat milk lattes, but why are we so obsessed with calcium levels in our skin? And like, I thought our skin makes calcium by itself. Like, what?
So basically, our skin barrier relies on homeostasis, which is a moderated by calcium gradient. This means that the amount of calcium naturally fluctuates as our keratinocytes migrate to the surface of the epidermis. It's a natural balancing act that's critical for our skin barrier's health.
Yeah, it sounds like quite the balancing act, and a really important one too, just since our skin barrier faces so many different stressors throughout the day, like UV radiation, there's like water loss, microbial invasion. So why would we need to actually know how much calcium we have in our skin?
So it's important to remember that calcium exists in 2 locations in our skin.
No way.
Intracellular and extracellular. Can you tell me which one is which?
Oh, I thought I was the one asking the questions here, but I can play along. So extracellular calcium sounds like it's located outside, like externally from the cells, and intracellular must be inside the cells.
A+, that's it. So yeah, when we measure the calcium content inside of the keratinocytes, we correlate a higher calcium content to a stronger skin barrier.
Nice. So measuring extracellular calcium, that wouldn't be like the same thing?
You're on the right track. The extracellular calcium content is harder to quantify since it's spread in between the cells. It's much easier to look at the amount of calcium contained in a cell. By focusing on the intracellular calcium concentration, we are more tuned into finding a cellular fluctuations and able to quantify the concentration with higher accuracy.
That makes sense. That's interesting. So how does this intracellular calcium measurement work?
So in the lab, we would start by growing the keratinocytes to confluency and complete media. Next, the cells would be individually incubated with experimental controls.
Okay, you say controls. What are the controls in this?
So there are 3 controls: a well with no treatment, just the cells in complete media; a well with the positive control, cells treated with calcium chloride in complete media; and a third well with the negative control, which are cells treated with EGTA and calcium chloride in complete media.
What are the controls used for? I know we have them, like you always see them, but like what are they used for?
Yeah, so experimental controls are used as the known variables. They set the standards and help us measure the percent change between the active ingredient's intracellular calcium concentration.
Okay, I see. So how does our active finally get into the mix?
After setting the controls, we can add the variable. For us, the variable is always the ingredient that we're testing.
Okay.
To the 3 wells, we add 3 set amounts of the active ingredient in complete media.
Okay, so that all makes sense. So what do we do next? We just let it all marinate, let it all season?
Pretty much, you could say that, yeah. So after the cells incubate for 1 hour, we add a cell permanent fluorescent dye, which exhibits fluorescent signals when excited at 488 nanometers. If it lights up under the microscope at that specific wavelength of light, it's an indication of intracellular calcium.
Oh, that's cool. That sounds high tech, like glow in the dark, but more advanced.
Yeah, pretty much.
So how do you get the dye to actually stay inside of the cells and not leak out and ruin the whole thing?
So we use a suppressant dye, which reduces the non-intracellular fluorescence. This dye also prevents extracellular transport.
Kind of sounds like a cellular pause button.
Yeah, exactly. So we can prevent the fluorescent dye from leaving the cell, so we can measure the fluorescent signal intensity generated by the dyed calcium molecules.
That sounds intense. Get it? Signal intensity?
Don't quit your day job.
That's a tough crowd. Anyways, after it's dyed, what's next?
So next, we put the plate under the microscope and we record the relative fluorescence of each well. Once we compare the fluorescence in the positive and negative controls to the complete media alone, we then establish the experimental model.
And now we can finally compare the fluorescence of the active, right?
Yeah, nailed it. So we can finally compare the active fluorescence to the complete media and calculate the increase in intracellular calcium levels.
Oh my gosh, I'm exhausted. That's such a journey.
I know.
Thanks for walking through that, but I'm exhausted.
Yeah, well, imagine how your keratinocytes feel, Meg. Consistently sensing calcium levels and maintaining skin barrier integrity 24/7.
It's definitely a big job for sure. I think I finally understand why this increase in intracellular calcium is so important to our skin's barrier. And this has really been a great episode, Grant. Thanks for joining us. I appreciate your time.
It has been great. Thanks so much for having me.
And thank you for joining us for another Active Concepts Masterclass episode. I'm Meg.
And I'm Grant.
And be sure to tune in next time for our next Active Concepts Masterclass episode.